So in class today, we started by zooming into the details of what is in a nucleus. So we have here a eukaryotic cell. Typically, when we look at a eukaryotic cell, the nucleus, it looks like it's filled with a whole bunch of uncooked or cooked noodles. How about that? It's just loose DNA that's able to get used, and it's called chromatin. So chromatin is the loose, kind of unorganized, non-replicated structure that DNA is found typically during about 90% of its lifetime in a cell. However, when we can actually see DNA, when the DNA becomes visible, that's only after replication. Replication is the doubling of the DNA content in a cell, and that happens in an interphase. And it usually ends up looking something like this, or this is the model that we use to demonstrate this. This is a chromosome. It's made of DNA and a sticky protein, or many sticky proteins called histones. It is one side that is an exact copy of the other side, and they are joined at a midline position by something that we locate and call the centromere. So left side and right side, as I mentioned, are exact copies of each other, but we call them a sister chromatid. So here we have one sister chromatid attached to the other sister chromatid at the centromere. This is our classic chromosome design. After we completed this 84C, I distributed this worksheet, which was a coloring worksheet, but I'm not super interested in students spending time coloring this, but I do want you to start analyzing it. Up here we have a cell's nucleus, only the nucleus. This rest of the cell has been removed. So in this nucleus, you can see one chromosome, but it's in that classic X shape. That means it's a chromosome and it's in an X shape because it is replicated DNA. That means left side, right side joined by a centromere. When you zoom into this actual strand, you'll see it's more like a really complex chain of DNA wrapped around very sticky proteins called histones. You'll see letter I is already labeled down here. We also took a segment and marked it with just highlighting the DNA in this section, and then we made certain that we understood that that DNA is going to be, we could just symbolically say that this particular length of DNA could be coding for one protein that could result in a trait. That segment of DNA, just that section of DNA without the histones, is called a gene. And any of the double underlined terms are going to be words that we want to make sure we understand for later. Down at the bottom, we made certain that we understood this sugar phosphate backbone just simply means the back side of a DNA molecule. If students want to do coloring, they can, but the words that we underlined here are really the most important. So backbone is a very important term, and it's actually called a sugar phosphate backbone. I'll explain tomorrow why that is. Histones we double underlined, chromatin and chromosome. And you can see the letters that correspond to those pieces. So we've got chromosome, chromatin, histones, backbones, gene here, again chromatin, chromosome, histones, chromatids, and then of course centromere.